Dear student, Model 10, Banding Techniques, Classification, Uses of Human Cytogenetics in Medical Sciences. The main objectives are to know about the history of banding techniques, to know banding techniques and classifications, to understand the molecular cytogenetic techniques, to understand the uses of cytogenetics in medical sciences. Introduction. Cytogenetics is the study of the structural properties of chromosomes and their behavior during cell division as well as their influence on phenotypes. It also includes the study of factors that cause chromosomal changes. In the 1950s, several technical developments were made. Chromosomes and genetics had little impact on the medical practice until the 1960s. The discovery of the chromosomal defect caused Down syndrome was responsible for the emergence of medical genetics as a clinical discipline. Progress in medical genetics from conventional karyotyping to the molecular karyotyping and gene mapping. Cytogenetics initiated the mapping of genes, which has led to the Human Genome Project and the complete sequence of the human genome. Now, genetic diagnosis is possible at the DNA sequence level. It is a remarkable journey from disease phenotype to karyotype to genotype. Therefore, cytogenetics has become an indispensable tool for the diagnosis of various genetic disorders, paving the way for possible treatment and management. With this background, let us learn the following in this episode. A. Classification of banding techniques. B. Staining and banding techniques. C. Molecular cytogenetic techniques. D. Uses of cytogenetics in medical sciences, and E. Conclusion. Identification and detailed analysis of human chromosomes with the introduction of chromosome banding became one of the essential innovations in cytogenetics. Based on the differential staining of chromosomes, the following classification of chromosome banding techniques can be made: G banding, R banding, C banding, NOR banding, Q banding. 1. Chromosome staining. Staining is used to enhance the visualization of chromosomes with various imaging techniques. Differential staining of a chromosome leads to the production of bands. This can be used to identify individual chromosomes, since each chromosome number produces unique bands. In this point of time, it is necessary to understand how stains binds to the chromosomes. There are several ways a dye can attach to a chromosome. A stain can link to the DNA through intercalations at minor groove or major groove binding or external binding. This depends on the type of interaction the stain have with the DNA, which can be either a covalent or non-covalent. Covalent interactions involve metal stains where the metal coordinates with the nitrogen atoms at position 7 of adenine or guanine of the bases of the DNA. This type of interaction results to major groove binding. Non-covalent interactions are hydrogen bonding, electrostatic interaction and pi-pi interactions. A stain can link to the DNA through several ways if it contains functional groups which can produce many types of interactions. Giemsa is a visible dye that binds to DNA through intercalation. Thus, it is used for staining of the chromosome. Giemsa stain is a mixture of cationic thiazine dyes such as azure B and anionic eosin dyes such as eosin Y. Chromosome staining involves the formation of a thiazine eosin precipitate in a 2:1 molar ratio. First two molecules of the small fast diffusing thiazine dye intercalate between the bases of the DNA in a configuration that favors eosin binding, which is the large and slow diffusing molecule. Therefore, the chromosome stain blue. Then the eosin molecule forms a precipitate with the thiazine molecules, causing the chromosomes to stain purple. The formation of this precipitate is favored on a hydrophobic environment. 2. G banding. G banding is the most widely and commonly used banding method for cytogenetic analysis that was first developed by Chi Bright in 1971. This technique is more potent in the stability and resolution of the bands produced. The visible light dyes are more stable and capable of producing brighter bands than fluorochromes. To produce banding, the chromosomes are pre-treated with a protease such as trypsin. We can also use other pre-treatment methods such as incubation of the chromosomes with hot saline citrate or treating the chromosome into a detergent or urea solution. These pre-treatment methods are used to extract a particular subset of proteins from the chromosomes. The banding is formed due to the differential extraction of the proteins throughout the length of the chromosome. This is due to the differences in the structure of the various chromosomal regions. The positive G bands or the dark bands corresponds to the hydrophobic regions of the chromosome that favor the formation of thiazine eosin precipitate. These regions are seen in the late replicating heterochromatin, which is characterized as condensed and rich in protein disulfide crosslinks. The heterochromatic regions are AT rich and are positive G bands. The negative G bands or the light bands correspond to the less hydrophobic regions of the chromosomes that do not favor the formation of thiazine eosin precipitate. These regions are found as the early replicating euchromatin with a loose structure and have their protein sulfur predominantly as sulfhydryl. The euchromatic areas are GC rich, therefore, they are with negative G bands. A study also reported that the role of the most tightly bound non-histone proteins in banding. The resolution of standard G banding can produce 400 to 500 bands per haploid genomes. This facilitates the identification of structural chromosomal aberrations of at least 5 to 10 MB in size. This resolution was improved after the introduction of high resolution banding based on the use of synchronized lymphocytes cultures by UNIS in 1976. Therefore, this technique was helpful to produce more than 800 bands in the pro-metaphase stages. 3. R banding. In this banding, the GC-rich euchromatin produces positive bands that correspond to the negative G bands. This banding is produced by incubating the chromosomes in an ionic solution at a higher temperature, followed by staining with Giemsa. This process causes the denaturation of the AT regions of the chromosome because the low melting point of these regions as compared to that of GC-rich regions. 4. C banding. This banding reveals the AT-rich centromeres that consists of constitutive heterochromatin. This technique involves the acid treatment, incubation in hot saline and treatment of alkali to the chromosomes. With these treatments, depurinate the DNA and breaks the DNA backbone. This causes the extraction of the DNA from some areas of the chromosome. Therefore, C bands are produced due to the differential extraction of the DNA. It is well known that chromosomal DNA in the C bands is more resistant to removal than DNA from other regions. The stronger interaction of proteins protects the DNA from extraction in the C bands than in the different areas of the chromosomes. 5. Nuclear organizing region NOR banding. This technique identifies the nucleolar organizing area of chromosome. NORs are located in the satellite stalks of acrocentric chromosomes and codes for ribosomal RNA genes. This is a simple silver nitrate staining technique for NOR banding that is useful in clinical practice to study specific chromosome polymorphism such as double satellites. 6. Q banding. Kasperson first achieved chromosome banding through fluorochromes in 1968 with the use of quinacrine mustard and aminoacridine dye. Then quinacrine mustard was replaced by a less toxic quinacrine compound called quinacrine dihydrochloride. The fluorescence of quinacrine quenches due to the amino group of the guanine bases of the DNA. Therefore, AT-rich regions of the chromosomes to fluoresce more brightly than the GC-rich parts. The bands produced are called Q bands. The bright yellow-green positive Q bands corresponds to the positive G bands. Thus, Q bands represents the facultative heterochromatin. However, quinacrine does not fluoresce brightly in the centromeric AT-rich region, indicating the involvement of other factors plays an essential role in banding. Besides, if guanine residues are present and spaced with high periodicity in AT-rich regions of the chromosomes, the fluorescence of quinacrine can be efficiently quenched. Non-fluorescence counterstain induces the proper banding with fluorochromes uniformly throughout the chromosome. This is also used to enhance banding patterns that do not have a very high resolutions. In situ hybridization and banding. For the first time in 1969, Purdue and Gall showed that the hybridization of satellite DNA probes to denatured chromosome in situ on microscope slides. This allows recognizing a range of chromosomal deletions, duplications and inversions. The chromosome banding improves the resolution of gene mapping and led to the characterization of aberration in cancer cells such as 9:22 translocations in chronic myeloid leukemia. Cytogenetics was once again contributing significantly to medical sciences. 2. Sister chromatid exchange SCE. SCE was made possible by incorporating BRDU instead of thymidine into replicating cells of 2-cell cycles. This helps for visualization of interchanges between brightly and dull fluorescent segments of sister chromatid. This was used to observe an increase of SCE in patients with disorders and cancers. 3. Fluorescence in situ hybridization, also called as FISH. In 1986, Pinkel and his colleagues developed the FISH technique. This permits specific nucleotide sequence in morphologically preserved chromosomes, cells and tissues. DNA probes are capable of hybridizing to complementary chromosomal regions and allows weaving the chromosome location of a particular gene or DNA sequence through a microscope. This produces a fluorescence dot at the chromosomal location where the labeled probe binds. FISH allows a higher level of resolutions than standard G banding approaches. FISH helps to establish the order of DNA clones relative to bands, breakpoints and analysis of nuclei in non-dividing cells to achieve a higher level of resolution. This can be used for the characterization and diagnosis of disease. For example, FISH analysis have also contributed to distinguish Angelman syndrome patients inherited the deleted copy of chromosome 15 from their mother, while Prader-Willi syndrome patients who inherited the deleted copy of chromosome 15 from their father. This is due to the classical case of chromosomal imprinting. A high resolution form of FISH is called fiber FISH, which is carried out using isolated chromosomes from nuclear architecture and exit as long stretched out DNA fibers, which resolves gene rearrangements and duplication with precision. 4. Spectral karyotyping, also called SCI, and multicolor FISH is also called as M-FISH. By using flow cytometry and the knowledge of the human genome sequence, cytogenetics have developed 24 color probes set which are used to label each chromosome with a distinct color. Chromosome specific probes are made by labeling DNA fragments derived from the entire individual chromosome with a distinctly colored fluorescence dye. These probes are pooled and used in hybridization experiment with metaphase chromosome spreads. Sky paints the whole chromosome in the same color, and the EMFISH uses various fluorescence dye to represent multiple painting probes at the same time. These techniques are the most powerful to identify small chromosomal rearrangements in individuals. 5. Comparative genomic hybridization is also called as CGH. CGH approach involves the isolation and fragmentation of genomic DNA from both control and experimental cases. The fragmented control DNA sample is labeled with green fluorescence, while the fragmented case DNA sample is marked with red fluorescence. These samples are pooled and used together as DNA probes in hybridization experiment with normal chromosomes. The green and red probes then compete to bind to the chromosomes. If the chromosomal regions are unaltered, the green and red probes should bind equally, resulting in an orange-yellow color. If the chromosomal region were deleted in the experimental sample, that region would appear greener under the microscope. While if the chromosomal region were amplified in the experimental group, the corresponding chromosomal region would appear red under the microscope. This helps to identify genomic alterations in the entire chromosome. Further, microarray based CGH methods are developed, which is the equivalent of conducting thousands of FISH experiments at once. The FISH technique is faster with better resolution than other cytogenetic tools. This identifies precisely the copy number and the breakpoints of segments which is lost or gained than does conventional CGH. One, to identify chromosome rearrangements. Chromosome banding is valuable for the identification of individual chromosomes and the identification of chromosomal rearrangements that have occurred in the course of evolution. Besides, this study leads to the understanding of the chromosome structure, and polymorphism of heterochromatic bands are useful tools for distinguishing between paternal and maternal homologs. AG-NOR staining can be used to identify the location of nucleolus organizer on chromosomes as well as to assess their activity. Identification of NORs in interface nuclei has prognostic value in various cancers. The banding pattern enabled the detection of various structural and numerical aberrations. This will help to identify carriers in healthy family members and of couples suffering from repetitive spontaneous abortions. The banded karyotype analysis combined with various molecular techniques lead to more precise detection of multiple syndrome in children. The microarray based study provides the ability to map the copy number changes to the genome sequence directly. to identify Philadelphia chromosome. In 1960, Peter Nobel and David Hungerford at the Institute of Cancer Research in Philadelphia uncovered a link between chronic myelogenous leukemia, which is also called as CML, and the presence of a small chromosome called the Philadelphia chromosome that was unique to CML cells. Later in 1968, Janet Rowley discovered the Philadelphia chromosome is due to a translocation event between chromosome 9 and 22. Then in 1985, Heinz Steenkamp and his colleagues mapped the breakpoint of the translocation and identified the fusion of parts of the BCR gene from chromosome 22 and the ABL1 gene from chromosome 9, resulting in a gene fusion product called BCR-ABL. The ABL1 half of the encoded fusion protein exhibits high tyrosine kinase activity, which is mainly responsible for CML associated phenotypes. In 2002, Drucker developed a drug called imatinib mesylate, also called STI 571 and Gleevec, to inhibit ABL1 kinase activity. Imatinib treatment of CML has been heralded as one of the biggest success story in cancer treatment over the past decade. 3. To diagnose Cray Du Chat syndrome and retinoblastoma. In 1963, Lugini and his colleagues discovered the association of Cray Du Chat syndrome, a congenital disorder with severe mental retardation, and a cat-like cry in affected infants with a loss of part of the short arm of chromosome 5. In the same year, Lilly and his colleagues identified association of the patients of retinoblastoma, a childhood form of retinal cancer, for the first official tumor suppressor gene RB1. 4. To map human genes. In 1965, scientists used human mouse somatic cell hybrid to map human genes to specific chromosomes. In 1968, Roger Donogh used the extended family pedigree and cytogenetic techniques mapped the Duffy blood group locus to chromosome 1. 5. To know cell structure. Flow cytometry and FACS were used to isolate populations of intact cells and individual fluorescently labeled human chromosomes to understand more about the cells and chromosome architecture. Advances in cytogenetics have made genetic diagnosis possible today at the molecular level rather than the phenotype. Cytogenetic techniques are bound to continue to be indispensable tool for diagnosis genetic disorders and suggesting possible treatment and management. Thus, this field will continue to uncover new, unexpected insights about both human genetic structure and genetic diseases. Thank you.